I am delighted to be talking today for the Wellness Brown Bag Lunch Series. I've been working with fatty acids, which are the components of lipids in the diet, since actually about when this cartoon was made, 1985. And I started out at MD Anderson in Texas, looking at how dietary fatty acids are helpful in preventing cancer, and there are certain ones that seem to be helpful in that, and it seems like I've just stuck with them for my whole career so far. So this is a cartoon that is sort of funny because I didn't intend to be working with them 25 years later, but here we are. So that's a cartoon that says it says little fatty acids, they've taken up jogging. And I have seen in the realm of science around lipids that there are circular kinds of popularity with different themes, and right now you're hearing probably a lot about omega-3 fatty acids, polyunsaturated fats in the diet, and so forth. We'll talk a little bit about omega-3 fatty acids at the end if you're interested. I do some work here at OSU with some other investigators on those. But today I'm actually going to focus on two other types of polyunsaturated fats that I'll introduce you to in a minute. Just in case you're wondering where nutrition is, it's that little dot that is right in the middle of, I'm sure I'm going to pull the computer down, but there we are. Campbell Hall is right there. There's Mirror Lake. The medical center's over here. There's a bunch of new buildings all around us, and then we're this nice old building called Campbell Hall. And we have a small unit there. Now it's not a department anymore. We've been Uber-sized into something different, but we still are a unit of human nutrition here at Ohio State University, and we do a lot of work in bench research, clinical research, and then even outreach and engagement with extension. So come by and visit any time, or give me a call if you want to look at labs and stuff. That's my group, basically, who has contributed to doing this work. A couple of key people that have done a lot of the work are Rachel, who is my chief manager here in the green sweater, and Kara Cleaver over there in the back. And Holly has done some of the work as well. And I have some newer people that aren't up there right now. But they're the people who are in the lab and in the clinic really working with patients to look at how dietary oils are helpful in health. And I'll show you some of those data as we go through the talk. So we're going to talk about how dietary oils fit into a healthy lifestyle. And I think I'll convince you that there are some really good benefits to including healthy oils in your diet. Very achievable, very affordable, and don't need to be actually used in supplements, although you may just choose to use some supplements. But some of the oils I'll talk about today are very readily available in supermarkets, and I'll tell you how to find those as well. I mentioned that I do work in cancer prevention, and I still do this work. And the bigger context of what we do now is we look at how dietary oils can impact metabolic diseases that predict for cancer, if that seems like a long kind of segue. We know that obesity is a big issue, and it's a predictor for reduced quality of life and decreased in mobility in people as we get older and heavier. It has a social stigma. We know people who are battling weight and dealing with being overweight not only have a social stigma all the way from childhood up through adulthood, but we know that wages are lower for people on average who are more overweight, and there's certain preconceived notions about people who are overweight. Whether that's true or not, it doesn't matter. And then we know that being obese increases the risk for many diseases, including metabolic diseases like type 2 diabetes and the metabolic syndrome, many cardiovascular diseases, as well as many types of cancers, including colon cancer and breast cancer, which is one that my laboratory works on. The theme that seems to be really coming up again and again in obesity and all of those kinds of diseases is this theme of inflammation. And we know in people who are overweight and have a lot of body fat tissue, adipose tissue, there's an increased level of chronic inflammation. And why we think that's problematic is that if you have a little heightened level of inflammation, it seems to predict for these diseases that are relying on inflammation to progress forward. And so if you walk around every day with a little bit more of inflammation in your body, it sort of is like the igniter for the fire for increasing your risk for the diseases like type 2 diabetes and heart disease and these certain types of cancers. So we actually do basic research in my laboratory, and a lot of our work is we look at how adipose tissue is responding to the diet and exercise. to not only shrink the adipose tissue in the condition of weight loss, but look how the adipose tissue is responding for things like inflammation. And this is sort of hard to see, so there's not a lot here, but there actually is a lot to us. We look at each of these outlines. This is an adipocyte or an adipose cell. So this is a very strong microscopic image of a section of adipose tissue that we got from a mouse. The mouse was very generous to donate it to us, and we're grateful for that. But these adipocytes actually look like just big circles. And the reason for that is the whole cell is actually filled up with a lipid droplet. So when we actually prepare these sections, the slides are very oily, and it's the oil from the oil that's stored in the adipose that's actually causing the slides to be oily. And adipose is important for all of these things here, and I'm pointing because I forgot my pointer today. But adipose stores oil, as I said, and the oil is used for energy, for making ATP, which is the energy we need to move around and repair our tissues and go about our day and keep ourselves alive. And oil is also a really good insulator. So we know adipose tissue keeps us warm. It actually is also a cushion for sitting on a chair. I mean, there's true physical insulation of adipose. And we used to think those were the only functions of adipose until about maybe 15 or 20 years ago, when we started realizing that adipose tissue also secretes its own hormones and other chemicals called cytokines. Some people call these adipokines because they're chemicals from adipose tissue. And several of them, I'll show you later today, we've measured some of these. Interleukin-6, which is a marker of inflammation. So it seems like the more adipose tissue they have, the more likely it's going to produce this interleukin-6. And this interleukin-6 has inflammatory effects in the body, some of which are good, but too much of which can be harmful. And then these adipokines are also secreted from adipocytes. And when they're secreted, they seem to regulate metabolism. So it's funny that the adipose tissue, which is a storage form for metabolism energy, also secretes things that regulate its own kinds of production of energy. So we've worked with people who have metabolic syndrome and type 2 diabetes, and almost always people who have those two conditions have central obesity. There's a definition for it of having a waist circumference of a certain width and so forth. But what we know about central obesity, in other words, the tendency to gain body weight in your abdomen region, we know that it's the best predictor of systemic inflammation that we know of. So if you have, without having other diseases. So if you are centrally obese, we know it increases your risk for heart disease, some types of cancers again, so you're seeing a theme here, Alzheimer's and metabolic syndrome and type 2 diabetes. And we look at inflammation and central obesity, especially in people relevant to the type 2 diabetes and metabolic syndrome. So that's really sort of the focus for the next couple of slides. So people who are centrally obese have what we call more of an apple shape of gaining body fatness. If they're going to gain weight in body fat, they're going to gain it more in their abdominal region. Some people call this male pattern obesity. And for women, we tend to gain more weight in our hips and thighs region, and this is called pear-shaped obesity. And it is pretty clear that actually for the same body fatness, someone with an apple shape is at higher risk for heart disease, cancer, etc. Whereas someone with a pear shape, even if they're obese, may not have the risk that we would see in the apple kind of comparative. What's really interesting too is when women go through menopause, it's not necessarily interesting, it's sort of sad, is we actually become more like apple shape when we gain weight after menopause. So what's been clearly shown is if you use hormone replacement therapy after you've gone through menopause, you shift your body fatness more back toward the pear shape. So it's clear that some of that is hormone dependent, and of course there are other issues with taking hormone replacement therapy, but that was one thing that was observed. And that sort of explains why after menopause women start to see higher incidences of heart disease, some types of cancers, type 2 diabetes, etc. It's that, we think it's that loss of estrogen and other things, and along with that, that male pattern obesity. So the metabolic syndrome, one of the key elements of it is being overweight, and especially abdominal obesity. And metabolic syndrome also has at least 2 other criteria: having impaired fasting glucose, having some kind of dyslipidemia, that's like high LDL or low HDL or high fasting triglycerides. And all of these things are measured in our screenings that we do here at OSU. All of these are actually every year. And then, or having high blood pressure. So any 2 of these, in addition to being overweight, which is the 3rd one, would predict, actually diagnose you as having metabolic syndrome. And importantly, that then is also a high risk, especially high risk for type 2 diabetes to eventually progress to. So my laboratory focuses on these elements of metabolic syndrome because of what I'll show you in terms of how we know some dietary oils seem to work. Right now, the best, I guess, or the most prominent way to treat metabolic syndrome is to help people try to lose weight. And that is certainly the best prescription, but it's not the most effective because we know many people who lose weight tend to gain back a lot of their weight, if not some of it, all of it. And that's, of course, a big problem in the United States. And so my laboratory is interested in finding other things that can help with traditional weight loss for managing metabolic syndrome and type 2 diabetes, and again, certain cancer risks. So this is the outline for what we will talk about today. And there's a little bit of chemistry, a little tiny bit, and I'll keep you wait through it. And then there's there's a little bit of, okay, wait a minute. And then we'll talk about what I think is new in the oil world. And of course, I love looking at oils since I've been doing it for so long. So maybe it's not news to you, but I'll tell you what's new in the world of oils. And then give you a little bit of idea of how we think oils are working in the metabolic syndrome and obesity research. And then finally, we'll talk about what we know and maybe what you need to know as you leave today. So yeah, this is the first slide of chemistry. And just really, really simply, actually the good news is lipids are very simple molecules. They actually just have a big carbon tail. This is called the hydrocarbon. They have this methyl group here. This is called the omega end. And the blue box down there is called the alpha end, or the carboxylic acid end. The alpha and omega come into play when we talk about omega-6 and omega-3 fatty acids. So I'll show you some of those in a minute. But basically, this is a saturated fat because all of the carbons are saturated with either other carbons or hydrogens. And we know saturated fats in the diet generally are solid at room temperature. This particular saturated fat is palmitic acid. There's 16 carbons in it, and no double bonds, and I'll show you what that means in a minute. And it's found in fats like shortening, all the stuff we're baking with between now and Christmas, or now and your holiday season is shortening, hydrogenated fats, maybe tropical oils, as in palm oil and other things. So saturated fats are in these solid fats that we use for good baking properties because they act as leaveners and tenderizers and very good for mouthfeel and other qualities we're looking for. So they're good, important. This particular fat is known to increase abdominal fatness in mice if they can be abdominally fat, which they can be. And so we think this particular fat, when consumed in excess, is likely to contribute to the apple-shaped kind of obesity. This is another saturated fat, so this is just 2 carbons longer. Otherwise, it looks the same. It's stored in adipocytes like the other one, the palmitic acid. It's called stearic acid, and I usually remember that. It's in animal dry foods because of steers being male bovine that are, I don't know, not quite male anymore, but sort of male. Anyway, stearic acid is an interesting saturated fat. It's about 1/3 of the fat in beef and dairy products from ruminant animals. It's actually not thought to be a cardiovascular risky kind of fat, whereas palmitic acid is, and that's sort of a detail you don't really need to remember. It looks very similar in the diet, though. It's solid at room temperature. It does have similar qualities for baking. Some people used to use this a lot in things like beef tallow. And it's with other saturated fats that can actually be a little bit more harmful for heart disease. So,
is this also like lard, like from pig or pork?
Yeah, pig is pretty similar in terms of its profile. And the other fat that's in lard and tallow and other animal-derived fats is this next one. Get to it. Let's see. About a third of the fat in animal foods is actually an unsaturated fat called oleic acid. We actually get most of our oleic acid in the United States, by the way, from beef and other things, and dairy, not from olives and olive oil. This is a monounsaturated fat, meaning it has one double bond right up here. So you can see those carbons are not hydrogenated or saturated anymore. And this end thing shows us where the double bond is, and you don't really have to worry about that. It's an oil then at room temperature, and it, as you know, an olive oil has some flavor to it that's actually attributed to some of the other compounds in the oil, not from oleic acid, but from some of the green pigments that are actually flavonoids and other things. So sources of oleic acid include what we consider the Mediterranean kinds of oils, olives and avocados, some nuts. And again, as I mentioned, in the United States, a lot of our oleic acid comes from animal-derived fats. Now, these are the fats that I work with, polyunsaturated fats. Now, when I say I work with them, I'm always working with all the fats because most foods have lots of different fats in them. So to really look at the diet, we look at all fats. But we focus on how polyunsaturated fats in the diet could have effects. And this particular polyunsaturated fat has two double bonds here and here. So it has 18 carbons, two double bonds. It's an omega-6 fat, and I'll show you this today, but if you want to talk about it more later, we can talk about it. The reason it's called an omega-6 is the first double bond is 6 carbons in from the omega end. And all of the omega fats have this kind of same nomenclature. You can actually figure out the structure if you know the classification, but you probably don't care about that as much as I do. But this actually was the standard for all of the vegetable oils in the United States until next year, which is where the oil companies are all actually deciding they're going to make it more oleic acid rich. And we'll talk about what that means later. But for now, this is the most prominent fatty acid in our vegetable oils in the stores that we buy vegetable oils. So things like corn oil, actually I have a little picture of some of those in a minute, so I'll give you those sources. This is an interesting fatty acid. It is essential. And in nutrition, when we talk about something being essential, it means your body can't make it from something else. It can't make it from other oils you consume. You have to get a certain amount of linoleic acid in your diet to be healthy. We realized this, and we saw this first in growing people and growing animals, where if they weren't given a diet with linoleic acid, they actually eventually would die. And now we know linoleic acid has a lot of other functions. It's components of every cell in the body. In phospholipids, that's the cell membrane that surrounds the cell. It's also important in forming a waxy barrier in your epithelial tissue. So your skin, which is a barrier tissue, is made up of a lot of waxes that have a lot of linoleic acid in it. So if you were deficient in linoleic acid, you actually show scaly skin and have other problems with your epithelial covering. And then linoleic acid itself regulates other fatty acids in terms of their metabolism. So linoleic acid has this kind of very powerful role to direct other fatty acids in terms of how they're going to be used in your body. So some sources of linoleic acid, all of these are crops that are grown in Ohio and in the United States. This is safflower oil, soybean, corn, sunflower. These are all naturally containing linoleic acid in high amounts. As I said, now GMO oils have become accepted in our U.S. oil supply, so I don't know how that's going to affect our supply. What's interesting about linoleic acid, and I sort of knew this when I started working in the area, but I didn't really know how old and how much people had studied it, was in 1957, which was before the year I was born, by just a few years. There were many papers actually published. Circulation is one of the American Heart Association journals, very good journal, and in that one, Dr. Paj and several other people had written an opinion paper saying the vegetable oils, the decrease in cholesterol is thought to be largely due to a polyunsaturated fatty acid known as linoleic acid. So you may, if you grew up around in the 60s or 70s or were sitting at a dinner table, you actually might remember, but a lot of people were consuming a lot of corn oil, Mazola corn oil, other types of vegetable oils as part of their dressings for salads and cooking. And it was really because there was a lot of push by the American Heart Association to consume enough of these omega-6 polyunsaturated fatty acids to prevent heart disease. The data were pretty strong. Many, many large studies in men had shown that men who had higher levels of linoleic acid did have lower heart attack prominence and incidence, and lower deaths attributed to heart attacks. So it was pretty clear that there was some kind of protective role. A lot of it, again, was attributed to this cholesterol change, but now we're, I think, from the work I'll show you today, knowing that it could be due to some other things as well. So, I'm a professor, so I have to have some questions for you, quiz questions. And I'll have 5 of them. So this is the first one. Fatty acids are small molecules that are stored in what tissues? Adipose, yes. Anything else? All of them? Yes, and the phospholipids are stored in all of them. And the adipose are also stored in those big lipid droplets, which are different than the heart cells in the heart and liver and muscle. But adipose certainly has a lot of fatty acids. So it actually is all of the above. A little bit of a trick question, I know. And I think I have one more, maybe 2 more. Adipose tissue functions to, you can see how I write. I really am. Are you either too tricky or not tricky enough? Yeah, so adipose tissue functions to store energy and insulate the body. We're going to talk about how it can alter gene expression by releasing those adipokines and IL-6, and it does influence inflammation, so it's all of the above. So my laboratory is really interested in how fatty acids in the diet really influence gene expression to change how other tissues are responding to the diet, and then body composition and metabolic syndrome and so forth. And we, about 15 years ago now, started working with a group of receptors called PPARs, and it's a long name. PPAR stands for peroxisome proliferator activated receptor, and I didn't name it, so I just call them PPARs. And what we know is the fatty acid, which could be that box there, binds to a protein called a PPAR, which is the yellow, and those two things together move into the nucleus and change the expression of genes. So all of you have inherited lots of different genes, all of the same genes, primarily 99.999%, we're all the same. And what we know is that the diet can influence how some of those genes are turned on or off, more or less. And we think that's in response to the diet because your body needs to regulate what it's going to use for energy. So what happens is when a fatty acid binds to a PPAR, it sits on a piece of DNA. So this is a string of nucleotides that make up DNA, and cause the DNA to be expressed into mRNA, which then goes on to express itself into protein. And the proteins that are expressed from those genes are regulating fat metabolism and energy metabolism. So that's sort of how we think dietary fats influence how fats in the body are used, is through this kind of genetic manipulation through PPARs. And there's quite a bit of evidence that that's in part how fats decide and dictate what your body is going to do with the fats once it gets in there. We know that a lot of the influences of fatty acids in the diet end up eventually changing adipose size, so the amount of adipose tissue, and other things like prostaglandins, which are involved with inflammation, and other genes that are involved with inflammation as well. And to give you an example of that, I'm going to talk about a few other fatty acids and show you how we compare fatty acids. So we looked at, this is the last chemical slide, I think. So we looked at linoleic acid a couple minutes ago, and it's that two double bonded fatty acid. It's in lots of vegetable oils currently. And my laboratory looks at 2 other fatty acids that are in ruminant oils, dairy products, cheeses from dairy products, and then beef and other ruminant meats. Primarily because we, many years ago, were looking at these in terms of metabolism, and we've just continued to do this work. And what we know is that the conjugated linoleic acids, those 2 other molecules on that fraction or on that slide, are in these ruminant products, and they're also marketed now as dietary supplements. And I'm not here to sell supplements because I don't don't own any, but the supplements are just showing you here. They look like oil supplements, and they're touted to be weight loss supplements. And so these have been on the market for many, many years, and my laboratory was interested in really making sure that they were doing what they said they were going to do. And so we actually set up a couple of studies that I'll show you some of the data for. We did some work in mice, and this is sort of a complicated study in terms of how this graph is, but this is body weight. The mice came in and we started them on their diets at about 32 grams. And in the solid, I'm sorry, let's just look at the circles. So in the black circle, you can see how the mice just gain weight over time. So this was a 30-day experiment. We fed the mice a controlled diet, and these mice actually are a very fat kind of strain of mouse. They gain weight very readily if you feed them a sort of typical moderate fat diet. But when we fed the same diet to mice in the circles here with CLA, that conjugated linoleic acid, we saw that it suppressed weight gain and even caused a little bit of weight loss. And that was a study that was published in the, about maybe 8 or 9 years ago now. And I won't go through what the pluses and minuses are. We tried to add a drug to look at some interactions of that, and that was an interesting part of it, but not relevant right now. Then we looked at adipocyte size. So we fed the mice for 30 days, actually in this case 17 days, and we looked at even a lower dose of CLA than used in that previous slide. But we looked at the size of the adipocytes, and you can see in the control-fed mice, the adipocytes are quite large. They're doing their job really well, storing a lot of lipid. When we fed the mice in CLA, the adipocytes were significantly smaller. We can measure this by having a graduate student spend hours and hours and hours behind a microscope actually measuring each little adipocyte. Important though for really showing what we wanted to show. And the CLA shrinks the adipocytes. So in mice it seemed to work out okay. We are fairly convinced that that was an important part of our finding. We also know that if you're shrinking adipocytes, the energy that's not being stored in these large lipid droplets has to go somewhere. And so one of the things we did was we looked at where else the lipid went. And to give you the short version of the story, some of the lipid goes to the liver for temporary storage. And some of the lipid is actually burned off as heat, which is called thermogenesis. It's how all of you stay warm. You actually produce some of your body heat through thermogenesis, producing heat. And so what we did was we measured basal temperatures in the mice, and this is the control diet. This is the CLA diet. And as you can see, the CLA mice had a significantly elevated body temperature. They went from about 33.2 degrees Celsius up to about 34.5 degrees Celsius. So significantly higher. We've actually gone on now to show how this thermogenesis is working, and it's an interesting story, not one that you might be concerned with too much, but it seems to be through causing some of the energy to be burned inefficiently as heat rather than making it into ATP. And so we're trying to understand sort of the energetics of how that's working in some of the work we're doing now with mice. But the other thing we wanted to do was to see if CLA could also work in people, since it's been used as it's been touted as a weight loss supplement. We wanted to see if it could work. And I was talking to somebody earlier today here in the room, and we were just discussing how, weight loss supplements are not regulated very well. We don't know what's in them. Actually, to a great extent, we sort of hope that what the package says is actually what's in them. And there's not a lot of proof that a lot of the supplements really do work. So we wanted to test it, and we tested it with an unrestricted gift. So we just got the supplements donated. We had some money donated for the study, and then we took the materials and ran with it. And so we designed a study to look at whether CLA could help people lose body fatness in the following study. And we did this in women. We called it the Women's Diabetes Study. It's a crossover study the way we designed it. So we had been on either CLA supplements for 16 weeks, or we compared them to safflower oil, which is rich in that linoleic acid, which we had known would be protective, maybe for some heart disease, things like cholesterol, but we didn't think would have any other effects. And we thought that would be a good comparative to look at weight loss and some other markers of metabolic syndrome. Maybe some of you were in this study actually, because we finished the study in 2008, and published a study first in 2009, and then we just published a paper last year. So if you're interested in that, or in it, let me know afterwards. When the women were on the supplement for 16 weeks, we then had a washout period where we took them off of the supplements and switched them to the other supplement. So what a crossover allowed us to do in this case was compare within the same woman how she responded to the CLA supplement or how she responded to the safflower supplement. It's a really powerful way to design a clinical study, but you can see how long the study had to be. It had to be 36 weeks total, which is a long time for these women, and they really worked hard for us to take these supplements. They took 8 of these pills a day for the whole 36 weeks if they were complying with our instructions. They also came in every 4 weeks, and we measured diet by doing 3-day diet records and other measurements, and of course body composition. At week 0 and 16 and 20 and 36, we did some more sophisticated measurements of body composition using a DEXA, which is a dual X-ray kind of imaging system to get a sense of body adipose and bone mineral density and other things. And we did a bunch of clinical measurements, fasting blood draws, and I'll show you some of those data as well. So again, we were comparing oils that were rich in these 2 CLAs in one group, or oil that was rich in oleic acid. That's sort of the big comparison there. So when we, this was a blinded study, double-blinded, meaning we didn't know throughout the 36 weeks what the women were on, and they didn't know. Only a third party who assigned the pills knew, the statistician. We had 55 women at the end of the study when we finished recruiting and running them through the study. They were all postmenopausal, all overweight, average BMI of 36. They all had had diabetes. We were looking at the interaction of the weight loss, hoping to see an effect on improving diabetes management. And they were all on various kinds of medications for their diabetes, different oral medications for lowering their glucose. Some actually were on more than one medication at a time as well. And so here, the big headlines are at the top, and I'll show you the data in a kind of compressed form. But basically what the 2 groups are showing you is whenever the woman would be on a safflower, either the first diet period or the second, this was the average effect that we saw. So when the women were on safflower, it looked like there was a little bit of an increase in body weight, but it wasn't significant from baseline. But when the women were on the CLA, their star here means that this lowering was actually significant, so that there was a trend for the data actually showing what really could be true. The p-value up in the top right-hand corner is saying that the 2 treatments were actually also significantly different. So when we compared how CLA worked versus how safflower worked in the same woman, we saw a significantly different effect. So 2 different kinds of statistics were done here. So CLA significantly lowered body weight, and it was about over a kilogram, which is just 2.2 pounds or 2.5 pounds if we add in a little bit decimal error. It's not something the women reported. They didn't really say they felt lighter after their CLA diet periods, but this was in a 16-week intervention, and they weren't doing anything else to change their behaviors. We asked them not to change exercise, not to start a new diet plan. If they changed their oral medications for the diabetes, they were supposed to let us know, and we believe they did. So we thought this was interesting, but body weight can change. We all know day to day with all of the things floating around right now between Thanksgiving and the holidays. So we also measure body composition, again, like I said, using some other sophisticated measures. And that's an image of a skeleton, as you see, but that's what kind of image you get from a DEXA or a dual X-ray observed diameter kind of instrument that we use for body composition measurement. And so what you can do from that is you can get an assessment from head to toe, basically of what someone's body, lean body mass is. That would be muscle, skeletal tissue, which is obviously calcified tissues like your bones and teeth, and then adipose, because those are 3 different kinds of density absorptions for the machine. We also can get an assessment of trunk fat, which would be in the rectangle from the hips up to the collarbone, basically would be considered trunk fat, which was our attempt at getting an assessment of central obesity. And just remember, every pound that is lost of adipose, at least, is a lot of energy. So when you're looking at energy balance equations, you got to figure out where the energy goes, which is always the question we get from reviewers, and for good reason. So CLA, these are the headlines. CLA lowered total adipose mass when we measured by DEXA. In contrast, safflower lowered trunk adipose, but did not lower total adipose, and I'll explain that in a minute, what that could mean. So when we look at total adipose mass, you can see CLA lowered adipose mass. So we believe, and we didn't have a section of fat to look at the cell size, but we believe CLA actually did shrink adipose depots in these women. Again, when they were on the CLA, we saw lower adipose mass. When they were on safflower, there was no change in adipose mass as measured by this instrument. And this was measured at week 0 and 16, or week 20 and 36, and it was measured both times for these women. Then when we just looked at the fat mass between basically the hip bone and the collarbone, safflower lowered trunk adipose by about 1.5 kilos, about 3.5 pounds of body fatness. So the women did report changes in their pant size or their bra size, because they would have included anything again in their trunk region. But we found a lot of body fatness gone when they were on the safflower treatment. This is the very first time we've ever seen this reported, but I have to say nobody's ever looked at how a vegetable oil can affect body composition, because I don't know why. But so this was a problem actually for us, because we didn't want our safflower to have any effects, and so we were sort of bummed about it. But it's exciting. Okay, so just keep this in mind. Safflower lowered trunk fat, but did not lower total fat. So we don't know if there was more fat in the safflower oil in the thighs, hips, arms, or where the fat was, but we do know it wasn't in the trunk. All right, so we looked at some other measurements of metabolic syndrome. How many of you know about triglycerides, cholesterol? You get them measured, right? We've talked about that. You get them measured here at OSU in your screenings every year. We looked at all of the other lipoproteins, and none of them changed from the dietary oils, except HDL went up by about 4 milligrams per deciliter, which is very good news. These women were all had lower HDL than they should. I think that's part of the metabolic syndrome profile. And CLA actually had no significant effect, but it looked like a trend downward, but again, it wasn't significant. We looked at measurements of inflammation, and this CRP is actually secreted when we secrete more IL-6, which, by the way, didn't respond at all to our dietary oils. CRP isn't secreted from the liver, and CRP was significantly reduced. So this marker of inflammation was down in our women. And we know CRP is actually up in people who have heart disease, again, and type 2 diabetes. So the fact that we reduced it was very exciting, and I haven't seen that with vegetable oils. We also know that these two adipokines are important in metabolism, and adiponectin was significantly elevated in the safflower oil group, and that's something we're pursuing, but it's not something you would get measured in a clinic, actually, right now. And finally, in these women, remember our hypothesis was that CLA would reduce body fatness in these women and could help them improve their glycemia. And you see that we didn't see an improvement. We really saw no significant effect. But safflower improved their fasting glucose by about 15 mg per deciliter on average. And HbA1c, which is a long-term measurement of glucose control, was also improved. So altogether, we saw quite a few benefits of this vegetable oil that is a pretty commonly consumed vegetable oil in the United States. Before you read the slide, remember that these women were supplemented with the vegetable oils, so they were consuming their normal diets. We didn't ask them to change their saturated fat intake, which is what most diet studies do. We didn't want to have that as another issue in our study. We wanted to just look at the effects of the supplement that we were testing. And so that's why we think we saw what we saw, is that it's actually truly an effect of the oil added, not something else that was taken out of the diet, which is always a problem in diet studies, is how do you interpret the removal of something versus the addition of something. So CLA did reduce body mass index and adipose, but had no effect on markers of glucose control or glycemia. safflower reduced trunk fat, markers of glucose control, increased HDL, and I didn't show you, but it increased lean body mass as well. So far, that's what we know about this clinical study, and we've published these data in a couple of journals. So here's the next couple of questions. Natural safflower oil is rich in saturated fat, monounsaturated fat, omega-6, polyunsaturated fat, omega-3, or CLA.
Let's see.
C, you think? Anybody else for C? How about E? Okay. Anybody going for D, A, or B? No, don't go for those. Okay, it's actually C. So we compared an omega-6 oil to CLA in that last study. So it was either one or the other. CLA in the supplements we use and the supplements that are on the market is actually synthesized in a laboratory. It's not even from ruminant products, but if you were to get it in the diet, it would be from ruminant lipids or ruminant oils. Omega-6 polyunsaturated fats are in safflower, corn, sunflower, soybean, those vegetable seeds that have a lot of oil. In our study in postmenopausal women with type 2 diabetes, CLA did which of the following? Decreased body weight in adipose, increased central adipose, decreased glucose or glycemia, all of the above. A, very good. Yay. Yeah, good. And finally, safflower supplementation did which of the following? Increased inflammation, improved glycemia, decreased HDL, increased trunk adipose, none of the above. B, right. Very nice. Okay, so here's the snapshot of what we think is happening. We think that central obesity, which is kind of this blanket risk factor for systemic inflammation, predicting for a lot of diseases, is part of how oils work to affect, we think, at least parts of the metabolic syndrome of diabetes. And we know from studies done 50 years ago, risk factors for heart disease and heart disease itself. We don't know a lot about Alzheimer's, which, by the way, is sometimes called type 3 diabetes, because there's some similarities to that disease, or cancers. And we don't study Alzheimer's in my lab, and we're starting to try to study some types of cancers. But here's what we think we work with in terms of my lab. I mentioned we look at how oils, or fatty acids in oils, interact with this group of receptors, these PPARs. And what's really cool for us to use as tools is drug companies have spent gazillions of dollars. That's a word that we don't hear in politics, but I think it's real. Gazillions of dollars to develop drugs that target the receptors that we know naturally oils actually already target and have targeted since probably the beginning of time. Some of these drugs are actually used for the metabolic syndrome and heart disease. So we use these drugs in my lab to compare to dietary fatty acids in terms of how these things work to interact with receptors. And again, we believe that what some of the fatty acids in perhaps in safflower oil do, and actually in CLA in a different way, is that they interact with the PPAR and change gene expression. And so a lot of the work we do to understand what's happening now with the women with type 2 diabetes is taking it back to looking at how changes in gene expression were happening and how that could change metabolism. And I'm going to move on from that and that as well. That's another cartoon just sort of showing you what we think happens with PPARs. Okay. So here's what we know. This is the last part of the talk, is that most people have heard of polyunsaturated fatty acids. About 77% of the population have heard of them. Maybe they don't know what they are, but they've heard of them. 21%, so of the 77, 21% believe that polyunsaturated fats raise the risk of heart disease. How many of you believe that? Not, hopefully not any of you after today, but they don't raise the risk of heart disease, okay? We also, most people actually know that omega-3 fatty acids lower the risk of heart disease, but few people actually know that omega-6 polyunsaturated fatty acids also lower the risk of heart disease. And I think this is published in 2010. I think what it's really was illustrating to me is that we see these waves of popularity in terms of media stories and science even about how different things in the diet affect disease. Right now, we don't talk a lot about simple vegetable oils as part of a healthy diet. We hear a lot more talk about fish oils, omega-3 oils, being important for inflammation, and they indeed are, but we need to also remind people about that particular easy-to-find vegetable oil. This is a label from, does it say what it's from? Safflower, yes, safflower oil. You can no longer get this oil, at least in the Giant Eagle I shop at, but this is an oil that is a naturally occurring oil with a lot of polyunsaturated fat, very little monounsaturated fat, and almost no saturated fat. And that's a typical vegetable oil that is a naturally occurring vegetable oil. As I mentioned, I don't know what the effects are going to be because I've never studied the oil before, but what's happening now is we're seeing a lot of oils with more like 11 grams of monounsaturated and 2 to 3 grams of poly out on the shelves, and it's because those oils are more shelf stable. The oil companies will promote them as being Mediterranean diet-like, but they're really doing it because it saves them money and allows them to store their oils longer. Anyway, the point of that is we don't know now what the implications of that are going to be. If you get vegetable oils, I recommend finding oils that have higher polyunsaturated fats on the label. And the way I'm finding them right now is I'm getting the generic brands for my household. So I don't promote any particular brand. If I could get a brand name, I would, but right now the brand names are all touting these they're called high temperature stable oils. They're also useful in frying so that you can use them longer in frying. But
can I just ask you a quick question about that? So you're saying that when you go in there and buy what's labeled as just
safflower.
Safflower oil,
it's no longer this one. It is, yeah.
Really?
And it's changed within the last year, yeah. It's amazing how the food industry. Okay, and the good news, they can do this, right? If this quality standard is now monounsaturated vegetable oil, they can do it. But what they'll respond to, of course, is consumer demand. So trans fats, you can hardly find oils and shortening with trans fats anymore. So they responded to the health research in that area. I'm an N of one out there saying, wait, don't get rid of all the polyunsaturated rich oil. Actually, there's a big group of people who are trying to continue to remind consumers that we need polyunsaturated rich oils because of the heart disease protection that we've known about for over 5 decades. But right now, you have to look a little harder to find the proof of rich oils. But they're cheap, they're easy to integrate into cooking and salad dressings and any recipe you want. They're very stable still. The polyunsaturated rich oils are stable. They're surrounded by antioxidants. That's how the seeds naturally protect the oils in the growing plants, so they're still there. It was really, truly a convenience marketing thing, I think, at this point. But, question?
Can you speak to the whole, omega-3 to omega-6 ratio in this idea that
yes.
we as Americans tend to get it a little upside down, and that's why?
Yes. So omega-3 oils, just really briefly, they usually have more double bonds than their omega-6 counterparts, relatives. We actually require this particular omega-3 in our diets as well. The thought is we don't consume enough long-chain omega-3s, which is not this one, but it is so this is alpha-linolenic acid. We probably get enough of that in our diets. We don't get enough of these two long-chain. These are omega-3s because you can see the nomenclature, but EPA and DHA. And the work I do with Jan Kiko-Glaser here on campus and Mary Freestad and Lisa Yee, some other physicians, are looking at how these two long-chain omega-3s could be helpful in reducing inflammation associated in the cases of those particular researchers, depression, anxiety, inflammation associated with depression, we're looking at some aging things associated with inflammation and then breast cancer risk. And we know that these two omega-3s are important for reducing inflammation. What the problem with the ratio, if you've heard of the omega-6 to omega-3 ratio, is it was intended to really help guide people to increase their omega-3 consumption more, not to reduce their omega-6 consumption. And unfortunately, some people have taken it to reduce their omega-6 consumption. I'm going to skip ahead here. By the way, good sources of those long-chain omega-3s are cold water fish. Some fortified foods, although some fortified foods have the shorter omega-3s, and those aren't going to be very helpful. This is the study. There's Jan up there. So the advice is to get your ratio of omega-6 to omega-3 in your diet 4:1. I don't even calculate that. I mean, that is complicated. But the advice was really intended to just remind people to consume more long-chain omega-3s, not to reduce their vegetable oil consumption. Unfortunately, some people have simplified the message based on what they want to people to think, that the omega-6s are pro-inflammatory, and they're not. I have a lot of evidence that they're not, and I've actually looked for evidence in the literature to show that they are, and I can't find it. Yes, you have a question?
I think most people think of omega-3s, they think of fish. So if you eat fish twice a week, what are the recommendations?
Yeah.
Will you be able to get this ratio of the omega-3s? Maybe not even talking about the omega-6s, but just
Yes.
will you be able to get your amount that you need?
Yeah, I don't
Sorry.
actually consult for the American Heart Association, but it is one of the organizations that has come out very, very profoundly and clearly that they want people to consume more omega-3s, and they advise most people to get 2 servings of cold water fish a week. It's exactly what they advise, unless you have a heart disease risk factor already, such as you have already been diagnosed with angina, you have dyslipidemia and hypertension or something like that. So if you do have that, they want you to have about 1 gram to 2 grams, which you really can't achieve with cold water fish very easily. I guess you could if you really, really eat a lot of cold water fish, but then they recommend going to supplements. 2 servings of fish a week will get you between 250 and 500 milligrams a day on average. Actually, the question you have is interesting. Lisa Yee is just finishing up a study where she's comparing supplements to salmon intake to try to approximate the same level, and we're trying to see if it accumulates in breast tissue and adipose and blood, and then if it has effects on inflammation. So we'll have more data hopefully coming out. So we don't know exactly, but we think it would help you get closer to achieving this ratio of increasing your omega-3 and not changing omega-6. So I think that's about all I had. I don't know what time it is because I
So
forgot
I'm
to
trying
check.
to make sense of everything. Bottom line is olive oil.
The
Our
bottom,
thumbs up.
olive oil is neutral, we think. We don't think monounsaturated
Everybody
oil.
thinks olive oil, olive oil, olive oil.
I know. Well, the data aren't there, unfortunately. It's not, they don't seem to be harmful. They're not harmful at all. They're probably but we know that linoleic acid-rich vegetable oils have protective effects for heart disease, and we think even some other parts of the metabolic syndrome. And that's been from decades of research from others, and just we added some new data on some of the components of metabolic syndrome that hadn't been looked at before. Nobody makes money off of saying go buy more vegetable oil, except maybe vegetable oil grows, but they're actually making a lot more money off of biodiesel like now. So that's another story. But it is a very achievable, affordable, easy to cook with. It doesn't have a lot of off flavors. You know, fish oil for some people is a little too strong for them to consume. I love it, but so.
And so my last thing, but then the saturated fats, just eat those on the lower end.
Yep, I mean, we're all probably going to consume more because of more baked things right now. They're just everywhere. I bake with them because you can't really get rid of them in some baked things. But that's not the oil and the fat that people are eating 365 days a year, really. Probably in this room. I'm talking to the choir, right? The audience outside of this room, they could be eating a lot of those fats on a lot of processed foods like chips, baked products that are packaged. The more saturated, the more stable the fat is too. So it not only adds some of the baked stuff that we like in home-baked goods, but it also is stable on the shelf. So, yep.
Is there, dose is probably too strong of a word, but like in a typical diet, what would be the amount or the serving of polyunsaturated?
Yeah, that's a really good question. I had some slides on that and I took them out. Our women still consumed with what we would call the RDA. So even though we added 8 grams a day, that's about a teaspoon and 2/3 of oil a day. It's really very minor. They also consumed it at each meal, so they had a little bit at each meal rather than all at once in the morning. We're trying to actually write a grant to do a study to look at dosing regimes and even providing it in different foods. Of course, nobody wants to fund that right now in the NIH, but we're trying to go to some agricultural commodities for that. So we think the amount is achievable with just adding maybe a teaspoon in the morning, a teaspoon at night, or even less than that. Do you want to buy supplements? You can. Actually, safflower oil is now packaged in a supplement. These people are making $1 million off of it because of my research. But anyway, I'm not a part of it. But I would just recommend trying to find a PUFA, polyunsaturated fat-rich oil, and making sure that you use it instead of saturated when you can.
Is flaxseed a polyunsaturated?
Flaxseed is a polyunsaturated with alpha-linolenic mostly, but it has some linoleic as well. And we frankly don't know if it's the antioxidants or the linoleic itself, so I would say that would also be okay.